Well, hello everyone, and welcome to the Malvern Panalytical web seminar program. So my name is Rob Taylor, and along with my colleague today, Carmen Wong, we'll bring you today's webinar, which is entitled Solid Form Control of Drug Substance, Ensuring Robustness During Scale-Up. And this will be delivered by Doctors Jen Morton, Pantelis Zydius, also known as Leon, and Carmen Gaguta. Just a quick note, this session, as usual, will be recorded and accessible from our on-demand portal on the website. A couple of other housekeeping points to notice as well. So on the right-hand side, you'll see a chat box entitled Poll Questions. So please click on that link there and go ahead and answer those poll questions. And this will run throughout the webinar. And if we have time at the end, we will attend to those answers and see if we can see any interesting trends that emerge. So please go ahead and answer those questions. Another chat box as well is the Ask the Speaker one, which we'll see above there. Please ask the questions at any time during the webinar. And again, we will try to attend to those if we have time at the end. Right, so before I introduce the speakers, I just wanted to give a brief context as to why we're running this webinar. And what you're about to see today is a nice case study as to how three organizations have come together to solve a problem with a recrystallization process, in this case with polymorphs. But I think what makes this interesting is how each organization has come together. We've shared knowledge, expertise and technologies in certain areas, and ultimately to solve this common problem. And of course, we've taken all these learnings and we've deployed that through Concept Life Sciences. So for me, this is a real positive example of how you can use collaboration as a means to drive innovation. So what I would always recommend is that if you have a problem that you can't solve in-house, look external, because particularly where you have scientific problems, then most often you'll find like-minded scientists who are very willing to help if you just ask that question. So with that, I'm going to introduce the speakers. I'll run through the biographies and then we'll bring the first one on. So up first will be Dr. Jennifer Morton. She's a synthetic organic chemistry at Concept Life Sciences. And after completing her PhD at the University of Leeds, she joined Peakdale Molecular, which is now Concept Life Sciences. And she has worked at CLS on numerous chemistry projects for clients. And now Jennifer concentrates most of her work on process research and development projects and has particular interests in developing re-crystallizations using design of experiments for reaction optimization. Up as well will be Dr. Pantelis Zydius, also known as Leon, as I mentioned. He obtained his PhD from University of Glasgow and continued his research in metal organic frameworks as a postdoc, after which he specialized in solid state and physical characterization. Leon was then able to move into contract research, where he is now a senior analyst, and of course is now focusing on APIs. And then finally, Dr. Carmen Gaguta. So after graduating with a PhD in solid state chemistry, Carmen then joined Avantium Pharma in the project leadership team. And then shortly after that, she joined Technobis, also in the project leadership team, before moving into business development, where she is now the global head. So with that, I'd like to bring on our first speaker, Dr. Jen Morton. And to you, I hand over the floor.
Lovely. Thanks very much, Rob. So today's talk is entitled Solid Form Control of a Drug Substance Candidate. First of all, I'll give you a brief introduction to Concept Life Sciences. Then we will talk a little bit about polymorphs. I'll give you a bit of background to the case study. Then I will hand over to Leon, who will talk to you about Xray powder diffraction and its role in monitoring polymorphs. We'll then hand over to Carmen, who will tell you about Technobis and the Crystal 16, which we used in this work. Then we will tell you more about the case study the solubility and the polymorph investigations that we've carried out. I'll then hand back to Leon to tell you more about the further physical characterization work that we did, and then we'll have final conclusions. Okay, so Concept Life Sciences. We are a contract research organisation specialising in pharmaceutical research and development. We have over 250 scientists working at 5 sites across the UK, and we support our clients' drug discovery programmes right through from early stage screening through to GMP drug substance manufacture. We have 2 chemistry sites, Chapel-en-le-Frith in Derbyshire, where I'm based, and Sandwich in Kent, where we work on chemical synthesis for all stages of the drug development pipeline. And then we have our site in Bradford, where Leon's based, where they focus on analytical work and, key to this presentation, the physical characterization work. We also have 2 sites in Scotland. They focus more on our biological services. So this slide starts to give you a little bit of an overview of what we do as an organisation. We, as I said, we work through the whole drug discovery pipeline, right from hit identification, computational chemistry screening kind of stuff, through to traditional medicinal chemistry work, and then through a series of more biological services. Moving on through the pipeline, we do scale up and process research and development and API characterization. And this is really the area where our talk today sits. We then have a range of further characterization and analytical services that are needed before you can move into API manufacture. Okay, so we are going to start off by talking about polymorphs today. What are polymorphs? Polymorphs are different packing arrangements of the same molecular species. I've got an example on this slide, which is paracetamol, and you can see images of 2 different polymorphs. In form 1, you can see that the molecules are sitting in a sort of a chevron arrangement, whereas in form 2, it's much more linear, got layers of molecules stacked above each other. Now, sometimes polymorphism is induced by tautomerism, hydrogen bonding, or other conformational changes in the molecule. But very often it's none of those, and it's just a packing arrangement of the molecules. It's important to note that hydrates, solvates, and salts are other types of solid form and can exhibit polymorphism in their own right. So a parent compound and a salt are not polymorphs of each other, but they can both exhibit polymorphism. Okay, how do polymorphs impact API production? The subtitle for this slide probably should be, why do we care about polymorphs? We care about polymorphs because whilst their chemical analyses and properties, like NMR, can be the same, the physical properties can differ. The properties that are going to have an effect on a potential drug are ones like solubility and stability. So the flowchart I'm about to show you is an extract from the ICH guidelines that define how we set specifications for drug substances. And you can see that we have to carry out polymorph screening. And if different polymorphs are observed, then we have to carry out further investigations. We have to prepare the polymorphs and characterize them and carry out physical tests and then move on to biological tests. And if we think that the different solid forms can alter how the drug works, then we have to go on to set a specification for polymorph composition in the drug substance. So how can we control the polymorph? Very often, the final stage of a drug substance synthesis will be a recrystallization, and a recrystallization gives us an opportunity to control the polymorph. This diagram shows the condition of a solution, how the condition of a solution changes with temperature and concentration. In the stable zone, we have enough solvent to fully solubilize the material. The material's happy in solution, and there's no need for any precipitation. In the pink zone, the unstable zone, where the concentration is higher, there's not enough solvent there to keep the material fully dissolved. And that means that the material can start to spontaneously precipitate. For chemists, that's crashing out. In the middle, we've got the metastable zone. And this is the zone where typically we would try and seed the solution. So the solution can persist in the metastable zone, but you can trigger it to start precipitating. So this is where we would like to seed, and the conditions here are perfect for crystal growth. So for good control of a polymorph, we want to see more crystal growth than nucleation. And the polymorph that can grow the fastest under those conditions in the metastable zone is the one that will dominate. And I think that Carmen will come back to this diagram later on and start to explain how we can find out where these solubility curves lie for a given solution. Okay, moving on to the background then for our case study. As a CRO, most of the work we do is client work, and in this case, a client had requested that we prepare a potential new drug substance, active pharmaceutical ingredient. So we inherited a medicinal chemistry route for an analogue, and we started by route scouting around that. Having demonstrated the synthesis of the target molecule, we moved on to scaling up the route with optimization work being carried out in parallel, which is something that we often do for speed. The client was keen to assess the solid form early on in the process, so we began looking for polymorphs as we were starting to develop the final recrystallization. And the goal was to consistently obtain one polymorph. So this is kind of a flow chart of where we got to in the client work. So we'd isolated a small amount of the drug substance to prove the synthesis, and we then began our scale-up work with PR&D being carried out alongside. We started to develop a final recrystallization process. We did a number of different trials with different solvent systems, and we isolated solids from those. We sent those solids over to Leon's team, and they identified 4 different potential polymorphs using xrpd. We then selected one of our recrystallization conditions, and we moved on to use those conditions to prepare a reference batch. Now, those conditions involved an antisolvent recrystallization, where you dissolve the material in a solvent it's happy to be in, and then you add a solvent that it is less happy to be in, and then having changed the conditions later on, it will precipitate out. So we isolated our reference batch, and that came back by xrpd as form 1, which matched that particular trial. So we carried on to continue our scale-up work on the synthesis, PR&D alongside again, and we put various batches of our potential drug substance through the final recrystallization, going up to 32 grams. I think we probably did about 4 recrystallizations here. Each time we obtained form 1. So far, so good. And then we brought the rest of our material through to the end of our synthesis and put that through the final recrystallization. So 74 grams, just over double what we'd done before. Isolated the material, chemically nice and pure, but when our xrpd came back, we had a different form. It didn't match what we had got before. Not only didn't it match form 1 that we were hoping for, it didn't match any of the forms that we'd identified at the beginning. So At this point, the client work moved in a different direction because this uncertainty about the polymorph was problematic for them. But we remained curious about whether we could gain better control of this process and started to think about how we could better understand the factors affecting the solid form in this recrystallization process. Okay, so I'm going to hand over to Leon now, who will talk about XRPD, and will show you some of the diffractograms that we obtained from XRPD here.
Okay. Hi, everyone. So, as Jen said, the basic instrument, the basic method that we're using is Xray diffraction. What is Xray diffraction? Just a brief introduction. Diffraction is practically the interaction of a wave with matter. How a wave bends around matter, like, for example, when a sound wave meets a wall, or when even things like waves bend around rocks on a shore. And this is, if we consider that our crystalline structure is a repeatable set of objects, in this case the atoms, and we employ the use of X-rays that have a wavelength of approximately 1 angstrom, which is close to the diatomic distance between the atoms in the crystalline structure, then that makes it appropriate for structural elucidation. How does diffractometer work? So first of all, in order to understand the X-rays interact with electron density around the atoms. When inside the powder sample, when we're talking about powder X-ray diffraction, we can assume that all those particles are small crystallites if it's very well crystalline. Those atoms occupy what we call as crystalline planes, as diffracting planes within the crystallite. And how this works is the X-ray beam from an X-ray source, when it hits at certain angles those different diffracting planes, and reflects, as we say, it's not really reflection, but it is called reflection in X-rays, and is detected at a certain angle from the detector. Then we get a signal, and all those signals from the different diffracting planes creates the diffractogram. That diffractogram is very characteristic of the phase that is within the structure, within the sample. And in order to get a representative sample and mitigate effects like preferential orientation or effects from different packing, etc., we usually tend to rotate the sample while it is being, the arms of the instrument are also rotating at the angles. And for those reasons, just because it is exactly very characteristic of the phase that is within the powder sample, makes X-ray diffraction practically the key physical technique in order to characterize the physical structural properties of APIs. It actually can be employed as a technique at all the stages of drug development, either if that is in the early stages, benchmarking what your physical form is, or in our case, identifying the different salts, the different polymorphs, optimizing the phase that we're going to be using, as well as identified how much crystalline our material is, but also identifying how much of one phase is within another, actually quantifying the phases if they're both crystalline or in whatever mixture within our sample. It can also be employed in later stages, like, for example, in quality control, batch release, identify the storage stability of the API as well as things like identified counterfeits or be used to characterize medical devices. On the right, I saw the instrument that we mostly used was the Empyrean S3 from Malvern Panalytical, and a really great instrument because of its modularity. Practically by changing the geometry of the X-ray source and the diffracting, detecting arm, you can practically change its abilities. Here I'm showing two different stages that we use during our measurements. On the top, on the right-hand side, on the top, you can see the reflection transmission stage, which is the basic stage. And practically here, you can see it in Bradford geometry. If it moves at 180, almost 180, it becomes a transmission stage. And on the bottom, it is a photo of the non-ambient stage where you can control the humidity and the temperature of the system while you're running your X-ray diffraction patterns. So to catch up from where Jen stopped, as she said, we identified 4 different forms, 4 basic forms, forms 1 to 4. Form 2 was not really a polymorph, which I'm just adding this up here for completeness. It would be strange not having a form 2. And in the end, we also found a form 5, which, as I will show you, it wasn't really a form. It was practically a mixture between forms 1 and 4. So after this, we assessed the materials with DSC, and we found out that we got really strange results. This prompted us to see what would happen after we would heat it up conventionally at 120 degrees. And interestingly, Form 1, that we had up until that point been considering that it being our basic phase, actually converted to Form 3. Here you can see the diffractograms from Form 1. With the red, it's the signal after it was heated at 120. And at at black is the reference diffractogram from Form 3. And I have here, I am presenting some of the most characteristic differences, the most characteristic peaks, but it's not the only ones. We can see the differences at the higher angles at approximately 30 degrees also. The same thing happened to that Form 5 that we isolated. And what prompted us to believe that it is actually a mixture of the two forms is the fact that after we heated it at 120, this time we got Form 4, which made us believe that probably Form 1 is formed and it is converted to Form 3 as we heat it up, whereas Form 4 remains stable after being heated up. So just to summarize briefly, this experiment showed that although we at first expected that Form 1 and Form 5 would be the basic and the most interesting and the ones we were targeting, nonetheless, the polymorphic landscape was more complex, and we were able to identify that probably we have 2 more forms, Form 3 and Form 4, which are more stable. And in order to gain a better understanding and a better control of this process, we would need to run some further experiments. Just to remind everyone that at this stage, we were very early on, and being able to identify the stable form at this stage is very important, as if we did it later on, it would cost quite a lot in resources and cost to repeat a lot of those studies, like toxicology studies or clinical studies. And with that, I will give the microphone to Carmen to give a brief introduction on Technobis and on the Crystal 16 and tell us how it works. And after that, then we'll continue with the solubility and the polymorph investigations. Thank you.
Thank you very much, Leon. And just before I start, I would like to thank Rob and Malvern Panalytical, and also to Jen and Leon for hosting this webinar and allowing us to join forces with you guys and bring this to our attendees today. I will not go into details because I also noticed that a lot of you that you are listening, joining us today, you are already a user of the Crystal 16 and Crystalline. I will get into the, just one second, I need to get a bit more acquainted with this. Okay, into the science. And before I would like to explain to you how it works, the temperature-dependent solubility determination in the Crystal 16 instrument produced by Technobis, I would like to take you one step backwards. And we all know out there are many, several methods on how to determine solubility, like, for example, the equilibrium methods, the temperature variation method, or polythermal method, how we call it, introduced by the Crystal 16 back in 2005 when it was brought on the market. And the solvent addition method is a method described a few years back by a group at CMAC, where they used the crystalline instrument of Technobis for multiple component systems. But I will not get into details there. I'll just go further. Many of you are acquainted with the equilibrium methods, with HPLC, gravimetric methods, are very accurate methods. But if you ever perform such a solubility determination, you also know that they are time consuming. Like, for example, if I think of the HPLC methods, you need to prepare your slurry, you need to slurry for 24 hours, 48 hours, depending also on company. Sometimes regulations are also different, and also on the compound, so you need to slurry for a longer time, develop a method. So it takes some time until you are able to actually get one solubility point for your compound in a solvent. And having this in mind, Technobis crystallization systems several years back, they brought on the market the transmissivity technology. And with that, the temperature variation method, or the polythermal method. It is a quite fast method, depending also on the compound and conditions. In a few hours, you can actually have a solubility curve, a few data points on your temperature-dependent solubility terms. And not only that, actually, you also have much more than that. In the same time, you can collect data related to the whole metastable zone. And I will show it to you exactly now how it works on the Crystal 16. You have a source of light, a LED light on everything. It's outside of the sample of the reactor, and this shines on your reactor and on the detector on the other side. So when everything gets into solution, when you have a clear solution, that moment is, of course, detected, and that's what you call your clear point. That's your first point on your solubility curve. And if you follow with a cooling, for example, type of temperature profile, and you are looking, of course, to the first moments when you crystallize something, so the light passes through, but when it crystallizes something, it's blocked. So you have also a decrease in transmissivity, and that's your cloud point. So actually what clear solubility light passes through completely, 100%. When something starts to appear into solution, I always am careful to say it crystallizes or not, because sometimes you can also make not necessarily a crystalline material. So to have a precipitate. And actually what you do, this is what you see also as an output from the software. So you have time here, temperature, and transmissivity on the other side. With orange, you have the temperature profile, and what you see with green and marked with blue, and it's actually the transmissivity with green. And your first, oh, I think I have, yeah. Animation, sorry. So your first point, so when you heat up your slurry, you get everything into solution, is the first point. When you have a clear solution, is your clear point, so your first point on the solubility curve. And if you follow with the cooling ramp, then the first point when you see something, so your transmissivity drops from 100% to 99%, so the first particles appear in the solution, that's your cloud point. And what we actually have in between the clear and cloud point is actually your metastable zone. So this is actually the real added value of this instrument to use it in solubility type of experiments, because you actually get much more from the data in one run, let's say. Simultaneously, you get much more out of it. And, sorry. Okay, so this is also what Jen has shared with you a little bit earlier. So actually what you see, all your clear points are the points on the blue curve, on the solubility curve, and the cloud points are the points that you get to define your supersaturation curve. So in the same time, you actually define the metastable zone with which actually most of us, we are looking for that metastable zone with, to find the right place where to produce later the ideal polymorphic form. And not only for that, but most of you, I think, also know that it's very important to define this metastable zone with well to maximize yields, to choose the right solvent, the solvent mixture, also the crystallization method. Even in solubility is quite important also for purity, getting rid of impurities, modifying or looking for the right particle size of your compound. So it is quite important. And with the Crystal 16, for example, and the transmissivity technology, you can get this quite easily with a little bit of material in relatively short time. You can get such data. There are many publications out there. Last year we had beginning of last year, so now it's we are almost 2 years further. We had a countdown, and there were more than 200 publications mentioning the determination of solubility, temperature dependent solubility with the Crystal 16, and as I already mentioned, why solubility matter is quite important. If you want to start a polymorph screen or any type of experiments that you start in the lab, it's very important to know solubility. And if you have access to temperature-dependent solubility, it makes things also even more easier to design your whole process or your screen. So as I have also here, so design of screens, crystallization screens, process, even if you want to make salts, co-crystals, you name it, solubility is quite basic. It's quite important to know and have a good idea of how your compound is behaving. And just before I give back the floor to Jen, I would like to show shortly a case study that we had not so long time ago by one of our students on ascorbic acid, the multiple solubility curves she made there actually determined the metastable zone within methanol, actually different alcohols and mixtures with alcohol and water. I think methanol, ethanol, and isopropanol. I will not get into details into these, but I would like to emphasize once again why it's so important to know well the solubility. The very first information that you can get on your compound, because as you can see for this case, depending on the alcohol you are using, or the solvent, or the solvent mixture that you are using, how much water or alcohol you are using, you would get a different actually crystal shape. In this case scenario, ascorbic acid has one polymorphic form, but you can modify the shape of the polymorphic form by using different mixtures of alcohol and water. But can you imagine how would be the problem if you'd have a compound that will be so polymorphic, and in each one of those combinations you will actually get a different polymorphic form? From one process point of view, I think this is also could be a very big headache. So this is get to know your compound well from very beginning, from the start, and as also Leon just mentioned, then you will save a lot of time, money later on in the process. This is just a bit to emphasize the many crystallization methods that you can do, and how you can design your experiments with our instruments, with the crystal breeder, for example, or the crystal 16. And just shortly, once again, to emphasize, to know what you are doing, those were the A and the B1, two case compounds that we had quite some years back, where what you see here, the different colors are the different polymorphic forms. So quite polymorphic also, if you think like that, of this compound, quite polymorphic compound. As you can see, if you would do only, for example, cooling crystallization, antisolvent evaporation, you would know 1, 2, 3 forms at the most. Here for this one, doing actually vapor diffusion type of crystallization, we really found out many other forms of this API, and for this one, I think the cooling evaporation, but a bit more diverse. So really get to know your compound by determining the solubility very early in your research, and then also it's much easier by introducing some diversity in your screens to get to know the compound. And now I leave it to you, Jen. I give it back to you.
Thanks very much, Carmen. Lovely. Okay, so now we will carry on with the case study work that we did using the Crystal 16. Just before we do that, I'm going to recap the relationships between the different solid forms as a starting point. So our first recrystallization and most of our recrystallizations through the process work up to 32 grams gave us form 1. And form 1, when it was heated to 120 degrees, converted to form 3. Our final recrystallization of 74 grams gave us form 5, which is the one that we believe to be a mixture of forms 1 and 4. And when heated at 120 degrees, that converted to form 4. So we're now in the position where we believe that forms 3 and 4 are more stable than 1 and 5. So what we hoped to achieve with this piece of work was we'd hope to generate solubility curves for forms 1, 3, 4, and 5. We had samples of 1, 3, and 5 retained, and we knew how we'd made form 4 previously, so we'd hoped we could make some more of that. We then wanted to compare and possibly overlay those solubility curves and see if we can tweak our recrystallization process for better control. We wanted to look at the effect that the antisolvent was having on our recrystallization. So that was something we had in mind that we wanted to investigate. And then if we've developed a new procedure, we want to go ahead and sort of test and verify that new procedure. This is the first piece of work that we did in the Crystal 16. And we did solubility curves for forms 1 and 5 here. So as I mentioned earlier, our original recrystallization process was an antisolvent recrystallization. But I'll come on to that later. And for most of our work in the crystal 16, we worked with a single solvent. We worked with just ethyl acetate. And that had been our good solvent in the antisolvent recrystallization. So here we have the solubility curves generated from the clear points, as Carmen mentioned, form 1 in red and form 5 in green. And then we've got the supersolubility curves over here with the metastable zone in the middle. What you'll notice about these curves is that they're really very similar, almost on top of each other, which was perhaps slightly surprising, but we hadn't done that. We hadn't done this kind of work before, so we just took what we obtained. We then moved on and we generated a curve also for form 3, and I will lay that over the top of those curves. So there we have the curve for form 3 in blue there. And again, you'll notice that they're all really very similar. So having done all these recrystallizations in the crystal 16, we isolated the samples, sent them to Leon's team for XRPD. And this is when we got our explanation about why the curves are all so similar. So if I move to the next slide, here are just a few of our XRPD traces. The red one is form 1 after it's been recrystallized from ethyl acetate in the crystal 16. The blue one is form 5 after it's been recrystallized from ethyl acetate. And at the bottom, we have our reference diffractogram from form 3. And you'll see that they are all the same. So at this point, we went back to our jars of form 1 and form 5, and we obtained new XRDs on them and confirmed that they were in fact still forms 1 and 5. They were stable in the jar, stable on storage, and they had both converted to form 3 during the recrystallization process. So the last piece of work we did in the crystal 16 was to look at our original antisolvent recrystallization. Now, we'd had a bit of difficulty repeating this exactly in the crystal 16 because once we'd added our antisolvent and the material had crystallized out, we were never able to get it to go back into solution. So we couldn't do the temperature cycling that you saw on one of the slides that Carmen showed. But we were able to dissolve our material in ethyl acetate, as we had done in the original process, and then add some antisolvent to the vial, put it back into the crystal 16 and continue with the cooling ramp and allow the crystal 16 to detect the cloud point. And the green line that you see on this slide is that data. It's made up from the cloud points of that run. And that is overlaid onto the solubility curves that were just obtained in ethyl acetate. I think that what this shows is how much we are narrowing the metastable zone with the addition of antisolvent. And I suspect that means that when we were doing our original recrystallization, we were actually very close to the supersolubility curve, and we might have been veering into the unstable zone and getting some additional nucleation happening. Maybe that is the cause of our reproducibility issues. Okay, so having done the crystal 16 work and obtained the solubility curves, which we now know are for form 3, we moved on to testing our crystallization. So we did some slightly larger experiments. We did 200 milligram experiments. We used ethyl acetate. We used a linear cooling ramp as in the crystal 16, and we took forms 1 and 5 and put them into a variety of experiments where we seeded at different temperatures. And in fact, an experiment where we didn't seed at all. And you can see that all of these experiments generated form 3 every time. We then went on to investigate the effect of the cooling profile. We took form 1 and we employed 3 different cooling profiles. So our original antisolvent recrystallization was probably running pretty close to a natural cooling profile where you have a steep ramp at the beginning. Crystal 16 uses a linear cooling ramp, and controlled cooling is something you hear about quite often in terms of recrystallization, where you have a very shallow ramp to start with and then it tails down later. So we ran experiments with each of those 3 profiles, and again we obtained form 3 from all of those experiments. We then wanted to do slightly larger experiments. We didn't have loads of material left, so these were half-gram experiments. And we used what we sort of considered to be our best theoretical conditions. We used ethyl acetate, we used a controlled cooling curve, and we seeded at 65 degrees C, which was at the concentration we were working at was approximately 25% into the metastable zone. So we took forms 1 and 5 and put them through our best conditions. And you can see that, as expected, at this point, we've got form 3 in quite a nice yield for a final recrystallization. The very final experiment that we did here was we wanted to see if we could replace our original antisolvent recrystallization with the new ethyl acetate recrystallization. So we repeated the final synthetic stage of our drug substance procedure to generate the crude material. And we did this experiment twice, and we attempted the recrystallization twice, and we tried to apply our new conditions to the crude drug substance. And both times it looked like it might be working. Chemists will be familiar, I guess, when you look at something and it's looking quite turbid and you think there might be some solid, and then you go to filter it off and it all goes to a bit of a nasty slump on the sinter. Oily solid, gum, and a low recovery. So what we have to conclude there is that our impurity profile is playing a large part in the success or not of this recrystallization. Okay, so to conclude that section, we have recrystallized forms 1, 3, and 5 from ethyl acetate. And that's led to the isolation of form 3 in all cases. We did a little bit of work to try and look at the impact that the antisolvent was having on the metastable zone. We have verified the conditions provided by the crystal 16 work, and we've investigated seeding temperature and cooling profile. We were not able to crystallize the crude material using the new procedure, leading us to conclude that impurity profile is critical. And therefore, we would say that the final purification of this drug substance is going to requires a 2-step procedure using the original antisolvent recrystallization for purification and then the new ethyl acetate recrystallization for form control. Okay, so earlier on in the project, when we had been focusing on forms 1 and 5, we'd done physical characterization work on those forms, but we hadn't done that work on form 3. So having generated now quite a lot of form 3, we took the opportunity to do that work, and I will now hand over to Leon, who will talk about the additional physical characterization work.
So hi again, thank you, Jen. So as Jen said, we got form 3. We had a lot of it, and it was a good time for us to benchmark these forms properties. We ran some basic characterization with the TGA, Nothing very exciting. There is one step, at approximately 257 degrees Celsius, as we can see from the thermogram. This is very typical. You have the decomposition of the material. Moving on, doing the same with DSC, we only see one transition at 127 degrees Celsius, which can be attributed to a melting event. Again, the one thing that we can take from these two is that we only have probably one form here, but further on, we suggested that we use our non-ambient XRD to examine the stability of the form. So the first experiment we ran a temperature-controlled measurement, ramping up from 25 degrees to 71 degrees at 50% relative humidity. That was close to the relative humidity on that day. There is no important change in the structure, in the form, with the increasing temperature. There is a slight left shift at the temperature as at the peak approximately at 70. It's more prominent there at approximately 17 degrees. This can be attributed probably to thermal expansion of the cell, and it is not great enough to change the phase, but you can also see it more significantly, this differentiation on the 3D isoline view, where we can see that there is a drop in the counts, which is normal. It happens when you are hitting a crystalline material, but it's not a great differentiation. We can say, though, that it remains stable at normal temperature conditions. But when we want to run some DVS measurements, we ran several of them. We had the material to do so. When we ran the DVS on the first cycle, we identified early on at approximately 10 to 20%, there is a small step on the isotherm plot. A small step that one of the possible explanations could be a crystal-to-crystal transformation. You could see the creation of a new hydrate, followed by a deliquescence step. On the desorption step, there is a hysteresis, and when the first cycle closes, it's not reversible. But the second cycle follows practically the system without that small step at 10 to 20% relative humidity. This step indicates that it is there is something happening at a lower humidity. Possible explanation would be that transition could explain why we got also so many differentiation between phases early on in the development. We also, of course, ran some XRD after the cycling of the material before and after. Here you can see the diffractogram. After the DVS, it corresponds to the form 3 as when you compare it with a reference diffractogram. Just some outtakes from this characterization. There's a lot to be done. We want to run a humidity-controlled measurement. Unfortunately, we are in the UK, and the synthesis was in the UK. We didn't take into account the humidity at those different measurements that we ran. Checking our Hanwell humidity systems, we have had humidities that range from 15 degrees up to 65 degrees, percent relative humidity up to 65%. And that's because we are in a tropical island like the Great Britain. And we haven't taken this into account, and probably this is also something we need to account for on further on the development. Maybe that's why for that reason a humidity controlled XRD measurement would elucidate this phenomena a bit more. And it seems that it is of paramount importance to control that, but at the same time it showed that consolidated form 3 as a target measurement. Also, it would be also very interesting, although we have cycled those measurements quite a few times, to run a more slow step TVS measurement. And I would pass right now for some general conclusions the microphone to Jen. And yeah, take it, Jen.
Thank you. Okay, so just our final conclusions to wrap up. We started with a recrystallization that was not providing a consistent, stable polymorph. We used the crystal 16 to generate solubility curves for a more stable polymorph, and the crystal 16 experiments and the subsequent robustness experiments that we did have only provided form 3. Experiments using the crude product have shown that we need a 2-step procedure in order to be able to control both the purity and the solid form of the material. We've done some additional physical characterization work on form 3, XRPD measurements to benchmark the thermal stability, and some DVS, which shows that we might have an interaction with humidity. And so further work on this compound will involve additional investigations. We'd like to do some variable humidity XRPD to fully investigate what's happening there. Okay, so that concludes our presentation for today, and just leaves me to thank the other people who've been involved in this work. Daniel did the crystallization, crystal 16 solubility work and the recrystallizations. Trisha, Gismon and Ronan helped out with the physical characterization work. Tilly and Caroline have kept us along the right track throughout the project, and Monsi has supported with project admin. So that just leaves me to thank Malvern Panalytical and Technobis crystallization systems for their technical support, instrument support, and collaboration during this piece of work. Okay.
Excellent. Thank you, guys. Thank you, Jen. Thank you, Leon. Thank you, Carmen. Excellent presentation. Carmen Wong, that is, would you mind closing the poll and then bring up the results? We only got a couple of minutes left, so whilst those are coming up, I'll ask a question that's come up in the chat, which relates to why would you expect a different result from the crystallization work if it's regardless of the initial form? I think this sort of relates to what if you're dissolving the initial form, what does that, why would you care so much over the initial form?
I think that's true. I think it's true to say that the form has no memory. It doesn't know what it was before. I guess at the start we were just experimenting, and we thought that because we were starting with conditions very similar to those that had given us, say, form 1, that we might get form 1 back again. That would have been my prediction, I think, at the beginning. I think what's interesting is that even the experiments we did with antisolvent in the crystal 16 generated form 3, even though we'd used exactly the same solvent system as we'd used to generate form 1. I think that builds on our purity profile argument. The impurity profile is affecting the solid form outcome.
Okay, thank you. Right, so we've almost run out of time here, but I think we can just quickly go through the results from the poll. So question 1 related to, have you developed a recrystallization step for a final compound? It seems that most of our audience did, of those who responded, so that's a positive response there. Question 2 relates to how often do you encounter that such let's say a problem with polymorph control, and the majority of people seem to did indeed have or encountered a polymorph problem. So it certainly seems that this topic is of relevance to our audience. If you can scroll down, thank you, Carmen. Do you use xrpd physical characterization? Yeah, well, it seems that the vast majority of people do, so that's good to hear, certainly from the Malvern Panalytical side of things. And then when it comes to turnaround times, well, most people seem to be able to turn around that within a relatively short period of time, so 1 to 3 days. So that's pretty good. But some folks have quite a long turnaround time, meaning longer than 2 weeks. So that's quite interesting in that respect, because it might indicate that you have an XRPD system somewhere off-site or in a different laboratory altogether. If you continue to scroll down. So how often do you characterize a metastable zone? So it seems that we've got an even balance here between, most people have either occasionally or frequently. So it seems, Carmen, that sort of relates to what you guys are doing. I think that, again, our audience is well placed for this particular topic. And then finally, the question on question 6 is, are you a Technobis crystallization systems customer? Yes, the majority of them are, so that's also quite good. Did you have any comments on those observations?
I would say that we have a very fast system. It is when it comes to the running experiments and running experiment for our customers. We have different turnaround times, but generally they don't go for several weeks. This is given to the fact that we have a lot of instruments when it comes to that, but just to boast also, we have a really high-end technology instruments, given the Malvern Panalytical, also Empyrean Xray. And any request, anything that you would like for us to measure for you, please do come to us. We can help you probably with that.
Okay, excellent, thank you. And it would seem that some of the audience as well don't tend to look at the polymorph landscape quite so much. So it seems there's a fair bit of opportunity there, Carmen, for you and your team to say educate some folk here.
Definitely, indeed. And quite a nice spread also in the poll what I see. So I guess also people involved more in the later stage process development and some in earlier. So quite nice bunch of people. Hope it brought the information necessary for everyone. And then let's make another one, Rob, for process, right? For later stage.
Yeah, that would be excellent.
Join forces also with Jen and Leon.
Okay, excellent. I think that we should probably draw a close to it there. We are 3 minutes over. So with that, I'll thank you all. Thank you to all the speakers once again. And also thank you very much to the audience for attending. So once again, to remind, this will be recorded and available on the Undermine website. And I think you'll also find links to it through the CLS and Technobis websites as well. So thank you very much, everyone, and have a good rest of your day.
Thank you.
Bye.
Bye.